morning to everyone. I'm going to talk about nutrigenetics and personalized nutrition. And I'm going to focus on three main areas of nutrigenetics. So the first part of my talk will be on importance of doing nutrigenetic analysis, why nutrigenetic analysis is so important, and why do we have to study gene-diet interactions. And for this, I'm going to give some examples from the Diogenes study. And the second part of my talk will be about why large samples are required to conduct nutrigenetic analysis. And for this, I'm going to give some examples from DCARDIA collaboration. And thirdly, implementation of nutrigenetics in developing countries such as India, Pakistan, Middle East, Morocco, and other developing countries. And for this, I'm going to give some examples from genuine collaboration. So before I go into my talk, I would like to give a brief overview about the gene discovery methods in genetic epidemiology. The first method that came into picture that was in early 1990s was a candidate gene approach, which is a hypothesis-driven approach that relies on the current understanding of the biology or the pathophysiology of the disease of interest. And what we do over here is basically we test for the association between a genetic variant and a trait of interest. And this trait could be any disease such as obesity or diabetes. However, this approach was not very successful because of two main reasons. One is that most of the studies were very small and hence they did not have sufficient power to detect the small effect sizes of the genetic variants. And secondly, most of the studies were unable to replicate the previous findings, which means that most of the candidate gene studies resulted in false positive findings. Overcoming these problems, in mid-1990, we had the genome-wide linkage approach, which is a hypothesis-generating approach, and this can identify a broad chromosomal region that harbors hundreds of genes associated with a phenotype of interest. Again, the phenotype could be obesity or diabetes. Again, this approach has got its own drawbacks. One is that, given that it's a linkage approach, it requires populations of related individuals, and that basically limits the number of samples that we could collect as part of the study. And secondly, it is unable to pinpoint the exact chromosomal location of the genetic variant. That is, with this approach, we will be able to identify several hundreds of genes, but we do not know which is the gene that is causing the disease. So overcoming all these problems, in mid-2005, that is exactly 10 years ago, we had this genome-wide association approach, which is another hypothesis-generating approach, and this can identify the exact chromosomal location of the genetic variant. And the three main advantages of using this approach is that it's cost-effective and it has got much greater resolution, and thirdly, it does not require population of related individuals. And it might sound that genome-wide association study is the best approach in the field of genetic epidemiology, but that's not true because several researchers are still using candidate gene studies in various fields of genetic epidemiology, and one such field is nutrigenetics. Nutrigenetics is basically gene-diet interaction analysis, and what does nutrigenetics refers to? It is the study of role of DNA sequence variation in response to nutrient. So I'm going to take the example of a study population, and I want to test whether a genetic variant is associated with obesity. And to test for the association of this genetic variant with obesity, and I see a very significant association. My next question is whether this association, is it a truly positive finding? So what I do, I look into all the lifestyle factors which can also affect obesity, such as dietary factors. So I look at the high-fat diet, how this could affect the association. So I stratify the population among those who consume high-fat diet and those who consume low-fat diet. And now I see an association with obesity only among those who consume high-fat diet, which means that not the entire population is associated with obesity, but it's actually 50% of the population who are consuming high-fat diet, they basically show an association with obesity. Which means that those who consume high-fat diet and those who carry the risk allele of the genetic variant, they are predisposed to developing obesity. So this is what we call as nutrigenetics or gene-diet interaction analysis. There's a contribution of both genetic factors and lifestyle factors which can contribute to the metabolic disease such as obesity. And there's another approach which is almost similar to nutrigenetics, which is called nutrigenomics. Nutrigenomics refers to the study of the role of nutrients in gene expression, whereas nutrigenetics focuses on genetic variants, whereas nutrigenomics will focus on gene expression. Now let's look at this example. Let's say we eat some food and the nutrients in the food, the nutrients could be micronutrients, macronutrients, any kind of nutrient. It enters into the body and it enters into the cell, and once it's inside the cell, it enters a cytoplasm where they are bound by different transcription factors. Depending upon the type of nutrient, if it's going to be a fatty acid, then there are certain transcription factors called as PPAR, peroxisome proliferator activated receptor. They bind the fatty acids. If it's going to be a nutrient which is more related to carbohydrate, then it would be carbohydrate response element binding protein. And if it's vitamin D, then you have the vitamin D receptor. So these transcription factors, they bind these nutrients and they transport them to the nucleus where you have the DNA, and they bind the DNA and regulate the gene expression. And the outcome of the gene expression would be the mRNA production, and which gets translated to proteins, and finally proteins are sent out of the cell to perform different functions. So when you look at the levels of mRNA, whether that is being modified in response to a particular nutrient, then you call it as transcriptomics. And if you look at the levels of proteins, how that is being modified in response to a particular nutrient, then it would be proteomics. Then you have the functional genomics and metabolomics when you look at metabolites. So all these omics approach will fall under the systems biology. But what is the purpose of doing all these omics approach? What do you think? Why do you want to know the levels of protein? Why do you want to know levels of the mRNA? Why do you want to look at different metabolites? What is the main goal? The ultimate goal is basically to identify the molecular biomarkers, which can help us to understand what are the mechanisms by which a particular gene is contributing to obesity in response to a particular nutrient. So the ultimate goal is the disease prevention. So nutrigenetics and nutrigenomics, the findings from nutrigenetics and nutrigenomics will help us to develop personalized diet, or otherwise called personalized nutrition. So nutrigenetics and nutrigenomics, which are two different approaches and parallel approaches, which basically leads to personalized nutrition. The nutrigenetics is basically the study of role of DNA sequence variation in response to nutrients, whereas the nutrigenomics refers to the study of role of nutrients on gene expression. So there are two important factors that play a key role in nutrigenetics and nutrigenomics. One is that there's a great diversity in the inherited genome. The genome in the Caucasians completely different from the genome in Africans. So there's a wide variation in the genome that makes you and me different from each other. So obviously this is one of the main challenges that we need to take into account when we do the nutrigenetic analysis. And secondly, people differ greatly in their food and nutrient availability, and this food choice depends on the cultural, geographical location, and also taste perception differences. This taste perception is genetically controlled. For example, if you take the food and nutrient availability in different parts of the world, in the UK there's increased consumption of high-fat diet. If you take Africa, there's a mixture of high carbohydrate and high protein diet. In Middle East, high carbohydrate and high fat diet. And Asian countries, you have increased consumption of high carbohydrate diet. So there's variation in genetic makeup, and there's variation in the dietary patterns. And obviously, these two factors are the key message for us to conduct these gene-diet interaction analysis. And what are the reasons for doing gene-diet interaction? Well, I say that we have to do gene-diet interaction because the dietary patterns are different, the genetic makeup is different. But what is the use of doing the gene-diet interaction? How is it going to help us? First, to test whether the modifiable dietary factors can overcome the effect of non-modifiable genetic factors on metabolic traits. Let's take the first example that I was explaining, where I showed that a genetic variation shows an association with obesity only among those who consume high-fat diet. So which means that the people who are consuming low-fat diet, they are prevented from developing obesity even if they carry the risk allele of the genetic variant. So which means that if you consider those individuals who carry the risk allele and you put them on a personalized diet such as low-fat diet, then they can be prevented from developing obesity, which means that the dietary factors can be modified, whereas the genetic factors cannot be modified. They are stable throughout your lifetime. So by modifying your dietary patterns, you can prevent the population from developing obesity. So that is one of the main goals of nutrigenetic analysis. And secondly, gene-diet interactions can mask the true effect of the genes on obesity. So let's go back to the same example. If I did not do a gene-diet interaction analysis, and I just stay with the main effect, that is looking at the gene and the obesity, then I would have reported that in this population, the gene is associated with obesity. That could be a misleading statement because not the entire population is showing an association with obesity. It is basically the gene-diet interaction that clearly shows the true role of the gene in obesity. So which means that gene-diet interactions can mask the true effect of the gene on obesity. So these are the two main reasons why we conduct nutrigenetic analysis or gene-diet interaction analysis. So what are the challenges in performing gene-diet interaction? It's not so easy because, first of all, genetic heterogeneity. We have already seen this. There's wide variation in the genetic makeup. The genome is quite different comparing Caucasians to Indians or Caucasians to Africans or Caucasians to South Americans. So there's a wide variation in the genetic makeup and insufficient sample size. I'm going to talk about this in the next 30 minutes, but briefly, large samples are required for conducting the nutrigenetic analysis. Gene-diet interactions will help us to identify very small effect sizes of the genetic variant. So when you do test for the association of a gene with obesity, then the effect can be very large. But when you test for the gene-diet interaction, there are three variables. One is the gene, one is the outcome, and then you have the dietary component. Three variables are involved, and when you test for the interaction, the effect sizes are going to be extremely small. So obviously, you need very large samples to detect the small effect sizes. And thirdly, incomplete coverage of the genetic variations within the gene. So for example, let's assume you take one gene and you're trying to test one genetic variant within this gene for the association with obesity, and you see that there is no association. You cannot report that the gene doesn't show any association with obesity because a gene can have hundreds and 200s of genetic variations, so you need to test all the genetic variation. But how can we test all the genetic variations within one gene? So there's one way to overcome this problem, that is this approach called as tagging polymorphism approach. There could be 10 or 12 genetic variations, and that represents the entire genetic variation within the gene. So if you genotype those 12 genetic variants, that will represent the entire gene, which is called as tagging polymorphism approach. So one way to do this is to capture all the genetic variation. That will explain the true role of the gene in obesity or any disease for that sake. Now moving on to the first part of my talk, which is going to be about the importance of doing nutrigenetic analysis. So I'm going to use the Diogenes study, Diet, Obesity and Genes, which is a pan-European program that focuses on the impact of genetic and dietary factors in relation to weight gain. And this study was done in 11,000 individuals involving 5 different European populations, Italy, UK, Netherlands, Germany, and Denmark. We tested for 2 effects as part of this Diogenes objective. One is the main effect, where we wanted to know whether the genetic variants were associated with obesity-related outcomes such as BMI, waist circumference, and weight change. And next, we tested for the interaction effect, where we wanted to know whether the dietary factors can modify the association between the genes and obesity outcomes. So we tested for 2 effects, the main effects and the interaction effects. And as part of this large-scale collaborative study, and we were involved in selecting the candidates, and this selection was based on several criteria. One was based on the Integragen, which is the diagnostic services company in France, which provided some of the important genes. And also based on the transcriptomics data, we selected some of the important candidates. And also the collaborating partners contributed their own candidate genes of interest. And independently, I carried out some literature search to identify important candidates. And at the time of the present study, there were few genes that were identified by genome-wide association scan. So they were included as part of the selection. So in total, we had 63 candidate genes. And as mentioned before, the main goal of this approach was basically to capture all the genetic variations within each of these 63 candidate genes. So we used the tagging polymorphism approach, and we selected 768 tagging polymorphisms from 63 candidate genes, which means that we tested for the interaction of these polymorphisms with different dietary factors on obesity outcomes. I'm not going to present all of these interactions now, but I've chosen a couple of interactions which are quite interesting. One would be the FTO gene, dietary factors, and obesity traits. Why FTO gene? FTO stands for fat mass and obesity associated gene, and the genetic variants in this gene have been shown to be consistently and strongly associated with obesity traits in various populations in different ethnic groups, in men, in women, in adults, and also in children. And these plots that you see over here, they are called as the Manhattan plots. They are produced as part of the genome-wide association scans. And the top hits in these scans were basically the FTO gene, which you could see for both adult obesity and as well as for the childhood obesity. So having seen such a strong effect of the FTO gene on obesity, the first question that comes to our mind is, can this effect be modified by a lifestyle factor such as physical activity and diet? Well, in order to answer this question, we carried out a study testing for the interaction between the FTO gene variant and physical activity on obesity risk in more than 200,000 individuals from different parts of the world. And in this forest plot, you can see that the overall p-value for interaction was statistically significant, suggesting that improving your physical activity levels can overcome the effect of the FTO gene on obesity. So having seen an effect of the physical activity, next we went on to test whether the dietary factors have got the same effect, for which we made use of the Diogenes study. As mentioned before, first we tested for the main effects. So we wanted to know whether the FTO variant was associated with BMI. And in this meta-analysis of the 5 cohorts, we found that there was a significant association with BMI, and also there was a significant association with waist circumference. So having seen that the main effects were statistically significant, next we went on to test for the interaction effects. We tested for the interaction with protein percentage, carbohydrate percentage, fat percentage, and glycemic index. And to our surprise, we found that none of the interactions were statistically significant. These numbers that you see over here, they are the p-values for interaction. And we found that the gene-diet interaction, that is the FTO gene dietary factor interaction, was not statistically significant. So on one side, we see that the FTO gene interacts with physical activity, but on the other side, we see that the FTO gene does not interact with dietary factors. So looking at this interaction between FTO gene and lifestyle factors in general, one could hypothesize that living a healthy lifestyle in general may not be sufficient to attenuate the FTO effect. In order to attenuate the FTO effect, physical activity seems to be the most important component rather than lifestyle factors in general. But however, this is very much of an hypothesis because of the fact that the gene physical activity interaction was conducted in more than 200,000 individuals from different parts of the world, whereas the gene diet interaction was carried out in just 5 European populations. So until we do the same, both the interactions within the same cohort of individuals, we will not be able to confirm this finding. Another example from the Diogenes study is the interaction between hypothalamic pathway genes and dietary factors on obesity, and we found that there was one gene which is called as the NMB gene. NMB gene encodes a protein called as neuromedin, which serves as a satiety signal and also plays a crucial role in appetite regulation. So we found a significant interaction with glycemic index on weight gain. So in this graph you can see on the x-axis you have the glycemic index, on the y-axis you have the weight change. These 3 lines, the 3 slopes basically implicate the 3 genotypes of the genetic variant. And as you can see, as the glycemic index increases, the annual weight change significantly increased for those individuals who carry the AA genotype, whereas the annual weight change did not change significantly for those who carry the GA or GG genotype. So this clearly shows that the AA genotype is a risk genotype. If you carry that genotype, which means that you're predisposed to develop obesity. So lowering the dietary glycemic index will be an effective way to prevent weight gain in those individuals who carry the AA genotype. This is one way towards developing personalized nutrition for those individuals who carry the AA genotype. And these are some of the other gene-diet interactions we identified in the Diogenes study. I'm not going to go into these interactions, but the main take-home message from this slide is that if you look at the main effects, you see that the main effects are not statistically significant in most of the situation, whereas the interaction effects are all significant. So this clearly shows that even if the main effects are not significant, it does not mean that the gene doesn't have a role to play in obesity. Maybe there are some hidden gene-diet interactions going on which are masking the true effect of the gene on obesity. So unidentified nutrient-gene interactions may mask the presence of a genetic effect in a certain environment or vice versa. And this is exactly why we do the nutrigenetic analysis or gene-diet interactions in order to understand the true role of the gene in obesity or any metabolic trait. So in line with that, I would like to thank all my collaborators for this Diogenes study. And moving on to the second part of my talk, why large samples are required to conduct nutrigenetic analysis. And for this, I've chosen the DCardia collaboration. I'm the lead study coordinator of this collaboration. And DCardia stands for Vitamin D Cardiovascular and Diabetes Related Traits. And to date, we have nearly 35 studies comprising more than 100,000 individuals from Western Europe, Scandinavian countries, North America, and Great Britain. And we also do collaborations with the consortia-based studies such as Giant, Charge, Global Lipids, and ICBP. And combining the consortia-based studies and the DCardia studies, we have a sample size of more than 155,000 individuals. But why have we initiated this DCardia collaboration? The main objective of this collaboration is to examine the causal relationship between vitamin D deficiency and cardiovascular disease related outcomes. So in order to examine this causal relationship, we used a novel genetic epidemiological tool which is called Mendelian randomization. In general epidemiology, when we test for the association between the exposure X and the outcome Y, the associations are highly confounded, which makes it difficult for us to examine the causal link between the exposure and outcome. So we use the genetic variants which mimics the exposure, or we use the genetic variants as instruments for the exposure. So the causality is inferred from the associations between the genetic variants and the outcome of interest. But why do we use genetic variants? There are two main reasons for that. One is that genetic associations are less prone to confounding, and secondly, they are free from the reverse causation, which are the two main problems in general epidemiological study design. So that's the reason why we use genetic variants as instruments in Mendelian randomization approach. So I'm going to give two examples from this. One is the bidirectional analysis, another one is the single directional Mendelian randomization approach. The best example for the bidirectional Mendelian randomization approach would be obesity and vitamin D deficiency. Well, we all know that lack of all these food supplements can lead to vitamin D deficiency, and excess of these junk foods can lead to obesity. And studies have shown associations between obesity and vitamin D deficiency, but what we do not know is the direction and the causality of the association between obesity and vitamin D deficiency. So in order to examine this causal relationship, we used a bidirectional approach because we don't know whether obesity is causing vitamin D deficiency or whether vitamin D deficiency is causing obesity. So we had to choose the genetic markers as instruments for this Mendelian randomization approach. So we have two exposures. When obesity is being tested for vitamin D deficiency, obesity will be the exposure. And when vitamin D deficiency is tested for obesity, vitamin D deficiency will be the exposure. So obviously, we need to select genetic markers for both the exposure. So we selected 12 BMI related markers for obesity and 4 vitamin D markers for vitamin D deficiency. And for statistical purpose, we combined them as BMI score, and 4 vitamin D markers were combined for as vitamin D allele score. So basically the question is whether obesity is associated with vitamin D deficiency. So we use the BMI allele score as the instrument for obesity and tested for the association with vitamin D. And likewise, vitamin D allele score was tested for the association with obesity because vitamin D score is the genetic instrument for vitamin D deficiency. If this is not clear, please do let me know. I'm happy to repeat it again. I know it's a bit confusing over here, but please do let me know. So we tested for these two associations. So first, testing for the association of BMI score with 25-hydroxyvitamin D concentration, which is the marker for vitamin D status. So in this DCardia meta-analysis, we found that the BMI score was associated with 25-hydroxyvitamin D concentration, where we found that for every increase in the risk alleles of the BMI score, there was a 6% decrease in 25-hydroxyvitamin D concentration. So this clearly shows that obesity leads to vitamin D deficiency, but we need to confirm the other side of the bidirectional analysis. So we also tested the other side, that is testing for the association between vitamin D score and BMI, and we found that there was no association. We initially thought probably this lack of association could be because of the small sample size. So we increased the sample size by combining the DCardia results with the Giant consortium, which has got like more than 123,000 individuals. But even after increasing the sample size, we did not find a significant p-value. So which clearly shows that obesity or higher BMI leads to lower vitamin D status, providing evidence for obesity as a risk factor for vitamin D deficiency, where we found that for every 10% increase in genetically instrumented BMI, there was a 4.2% decrease in 25-hydroxy vitamin D concentration. So we were able to prove this causal relationship. The next example that I'm going to give is a single directional Mendelian randomization approach, which is the vitamin D and blood pressure. Well, several studies have shown associations between vitamin D and blood pressure, but the findings have been quite inconsistent. For example, if you look at the epidemiological studies, they have shown a link between low vitamin D status and high blood pressure. However, the clinical trials have failed to show a link between vitamin D supplementation and hypertension. Even the largest epidemiological study to date, which was done in more than 12,000 individuals, again did show a link between vitamin D status and blood pressure, but failed to show a cause and effect. So we use this single directional Mendelian randomization approach to examine this causal link, and we use the vitamin D genetic score as instrument for vitamin D because we have just one exposure over here, and we tested for the association with systolic, diastolic and hypertension. Now first looking at the association of the vitamin D score with blood pressure, where we combined the results with the ICVP consortium to get a maximum sample size to increase the statistical power, and we found a significant association with systolic and diastolic blood pressure, where we found that for every increase in the risk allele of the vitamin D score, there was 8% and 10% increase in diastolic and systolic blood pressure levels respectively. And we also found an association with hypertension, where we found that for every increase in the risk allele of the vitamin D score, there was 1.02 times increased risk for hypertension among those who carry the risk alleles of the vitamin D score. So this clearly shows that using vitamin D genetic score as an instrument, we were able to show for every 10% increase in 25-hydroxy vitamin D concentration, there was 0.29 mmHg decrease in diastolic, 0.37 decrease in systolic, and 8.1% lower odds of hypertension. So this shows that improving your vitamin D levels can overcome the effects of cardiovascular disease-related outcomes such as blood pressure and hypertension. And this finding was quite recently published in Lancet Diabetes and Endocrinology. So in line with that, I would like to thank all the DCardia collaborators. So the main take-home message from this story from the DCardia collaboration is that we just tested for the association between the vitamin D score or BMI score with different outcomes such as obesity and blood pressure. And you could imagine that we had to get more than 150,000 individuals to identify very small effects such as 0.29 mmHg decrease in diastolic, 0.37 mmHg decrease in systolic. So only if you have such large samples, you will be able to identify such small effect sizes. So obviously, when you think about nutrigenetic analysis, where you have the gene, the outcome, and the dietary components, where three variables are involved and testing for the interaction, obviously you need a much more larger sample in order to identify that small effects involved in the genetic variants. So going on to the last part of my talk, which is about implementing nutrigenetic analysis in developing countries. So this is about the genuine collaboration. Genuine stands for the gene-nutrient interactions. The main objective of this collaboration is to perform large-scale analysis of gene-nutrient interactions on metabolic outcomes using population-based studies from various ethnic groups. So I'm currently developing these nutrigenetic analysis in different populations such as India, Pakistan, Morocco, Brazil, in addition to the ongoing nutrigenetic analysis in the UK population. But why am I looking at these different countries? So we need to look at the nutritional challenge. For example, if you look at the UK population, high-fat diet, Pakistan and India, high carbohydrate diet, Moroccan population, high carbohydrate and high fat diet, and Brazilian population, again, high fat and high carbohydrate. So you could see that the genetic makeup is quite different in all the population, and you can also see that the dietary patterns are different. But there's one thing that is common in all these populations. What is that? What do you think is a common thing in all that unites all these populations? Can someone guess? It's none other than the increased prevalence of type 2 diabetes and obesity. So we have different genetic makeup, different dietary patterns, but there's increased prevalence of type 2 diabetes and obesity in all these populations. So you don't need a better justification of why you need to do nutrient-gene interaction analysis. So I'm going to give one example based on the genuine collaboration that I did quite recently in collaboration with the Dr. Mohan's Diabetes Center in Chennai in India. And this is a collaboration between UK and India, and where we tested for the interaction of some of the important genes with macronutrient intake on type 2 diabetes and obesity, and we found one significant interaction, that was the interaction between the FTO variants and macronutrient intake on obesity and type 2 diabetes. Now looking at that particular interaction alone, well, for this interaction analysis, we used a study population from Chennai, which is based on the Chennai Urban Rural Epidemiological Study, in short called CURES, and which is a case control study where we had 1,000 individuals with type 2 diabetes and 1,000 individuals who were controlled, that is normal glucose tolerant individuals. And the dietary intake was measured using a semi-quantitative food frequency questionnaire. Now looking at the results of the interaction, we found a significant interaction between FTO variant and carbohydrate energy percentage on obesity. And in this graph, on the x-axis, you find the carbohydrate energy percentage in tertiles. On the y-axis, you find the odds ratio for obesity. And these bars represents the risk allele of the FTO variant. We found that the overall p-value for interaction was significant. And the conclusion was those who carry the risk allele of this FTO variant and those who consume high levels of carbohydrate, they had 2.64 times increased risk for obesity compared to those who do not carry the risk allele of the FTO variant, which means that any Indian who consumes high levels of carbohydrate and those who carry this FTO variant, they are at risk of developing obesity, and the risk is 2.64 times. We also found another interaction, that is the interaction between the FTO variant and carbohydrate percentage on type 2 diabetes. You see a similar graph over here. On the x-axis, you have the tertiles of carbohydrate percentage. On the y-axis, you have the odds ratio for type 2 diabetes. And the overall p-value for interaction was statistically significant. And we found that those who carry the risk allele of this FTO variant and those who those who consume high levels of carbohydrate, they had 1.66 times increased risk for type 2 diabetes compared to those who do not carry the risk allele. And this is the first nutrigenetic study in this Asian Indian population, and we have shown that the two FTO gene variants, they show a very significant interaction with carbohydrate energy percentage on obesity and type 2 diabetes. Now, by identifying those individuals who carry the risk allele of these two FTO variants, we will be able to provide them a personalized diet, such as a low carbohydrate diet, which will prevent them from developing obesity. And this is a way how you can take the finding from nutrigenetics to personalized nutrition. So I would like to thank all my collaborators from this genuine collaboration. So if anyone has got any cohorts with data on dietary intake or genetic data and obesity and type 2 diabetes, please do contact me. I would be very much happy to have you on board in this genuine collaboration. But before I finish, there's just one slide to explain how nutrigenetic findings can be translated to personalized nutrition. A lot of researchers are working on nutrient-gene interactions, testing for nutrigenetic analysis, and parallelly, we are also doing the nutrigenomics approach, doing transcriptomics, proteomics, metabolomics, and metabonomics, and functional genomics to identify different molecular biomarkers. So the finding from nutrigenetics and nutrigenomics needs to be translated, but there's one intermediate step, that is the genetic variants that show an interaction needs to be functionally characterized, for which we need well-equipped molecular genetic analysis. So once it is functionally characterized, then you could start developing the dietary intervention studies, which could be done based on the knowledge of the genotype. So we are currently doing some of the dietary intervention studies and also nutrigenomics analysis, but the question is about developing the personalized nutrition, how you're going to develop a personalized diet depending upon the genetic makeup of an individual, for which the key challenge is basically developing a database where you could integrate the data from nutrigenetics and different omics approaches and molecular genetic analysis, and that needs to be translated to personalized nutrition, and that is something questionable, and I don't know how many decades it's going to take, like in order to identify the methodology for identifying this personalized nutrition. And thank you all so much for listening to my presentation. I'm happy to take any questions.
Thank you very much for your nice presentation, Dr. Vimal. Now it is open for discussion. If there are any questions.
Okay. The FTO gene SNP is interesting.
Yeah.
Related to two different phenotypes. And I was just wondering whether the SNP is at the intron or exon, like why these genes are related to those phenotypes.
Yes, most of the genetic variants that have been identified in the FTO gene are from intronic region. And FTO gene was identified based on the genome-wide scan, and it is not a candidate gene, definitely. So the genetic variants were basically from intronic region, and till today, people are still struggling to identify whether these intronic variants are in linkage disequilibrium with the other genes outside the FTO gene, whether those genes are the ones which are contributing to these associations. So that's still not a question.
Yeah, almost like it's very likely that it is in the intron. Maybe it's during the recombination.
Yeah.
The whole thing flip over.
Exactly, but there's a possibility of being high LD with another gene which is actually involved in obesity or any metabolic trait.
And also, if a patient that's identified as the suspect, like as with this unfortunate genotype, they require, for instance, a low carbohydrate food. In that case, what would you suspect? What would you suggest? Would you suggest them to take? Because they require certain energy, right, in their nutrient. So would you suggest them to take more fat food to avoid carbohydrate, or what would be the suggestion?
Well, at the moment, I wouldn't suggest anything just based on one gene, because obesity is caused by hundreds of genes. So this is one finding based on one genetic variant.
Mm-hmm.
So you need to identify the entire panel of all the genetic variants.
Okay.
And only then, based on that, you're going to identify what personalized diet you're going to provide them. So it's not just based on low carbohydrate. So it's going to be a balance of carbohydrate, protein, and fat. Obviously, in that combination, the carbohydrate levels will be much lesser.
So if a patient have this kind of genotype, then you would suggest to test the whole
panel, yeah.
panel.
But still, we do not have the entire panel to date. Yeah. Because to date, like, we have only explained 2 to 4% of the genetic variation relating to obesity, which means that the remaining 96% of the genome is still unexplored.
Right.
A long way to go, yeah.
Relating also to this discussion, what about the role of microbiota, gut microbiota? Because we didn't hear anything about.
Yeah, of course. Yeah, we are also looking at the gut microbiota and looking at different hypobolome and metagenome interaction analysis. Yeah, that also, as part of my previous slide, that should be falling under the nutrigenomics analysis, which will include the gut microbiota as well. Yes, of course, that's a good point. Yeah, thank you.
And from you also. How far are we from the personalized nutrition, from the application? Because I'm also very interested in this field, but I realize that it's very complex.
It's very complicated, and quite recently at the University of Reading, we have developed personalized nutrition software, which is called as Food For Me, which is for basically those who have high levels of lipids, which is based on one genotype, that is the ApoE4, apolipoprotein 4 genotype, which is quite consistent in all the population. And we have published it. You can have a look at that website, and that will explain. But we haven't gone to the level of developing personalized nutrition for these multi-factorial disorders such as obesity or diabetes, because that's even more complex than lipids. Thank you.
Yeah, it's really intricate problem, because you are telling about the linkage between the low vitamin D intake and obesity.
Yeah.
Is there any relation between the geographically, because India, I think there is no problem with vitamin D, and in UK there is a problem, no? Vitamin D.
Yeah, the Western, yeah, of course. Yeah, when you do the vitamin D analysis, obviously you need to take into consideration of the ethnicity. But this D-cardia collaboration was done mainly in the Caucasian population. Also within the Caucasian population, we stratified them as Scandinavian countries and North America, UK, and Europe. The reason is, in the Scandinavian countries, obviously the vitamin D deficiency is pretty much higher. So definitely, yes, geographic location plays an important role, and we do adjust for all these confounders when we do all the analysis. Yes, what more?
Can this Nutrigenetics study be extended to Africa?
Oh, well, actually, we have just started a study in Ghana. And we are planning to implement nutrigenetic analysis, because I haven't mentioned all the population that I'm working on. I'm working on Ghanaian population, I'm working on Moroccan population, I'm working on the Middle East and Saudi Arabian population, and other population includes India, Pakistan, and Brazil. Yes, we are also working on the African population. Yes, of course, why not? Okay.
If there are no questions, thank you very much for nice presentation.
Yeah, thank you.